All right, so here we are. When it comes to red blood cells, first I want to talk about the lab values. So when it comes to red blood cells, what's normal? Okay, so if I take a low end and a high end, what are the normal lab values for red blood cells? So the normal values, for instance, so the way I remember this when it comes to red blood cells, I remember the number 5, right? And what comes before 5? Well, you have 4. And then what comes after 5? You have 6. So if I take 5, before that is going to be 4. After that's going to be 6. So I have 4, 5, and 6. Everybody with that? Okay. When it comes to red blood cell count, the normal lab values should be, it's not exactly 4, it's about 4.5 million. 4.5 million to 4.5 to 6 million red blood cells per microliter. All right, so let's write that in for you. I said when it comes to red blood cells, we should have anywhere from 4.5 million to 6 million red blood cells per microliter, okay? So that's going to be 4.5 to 6 million red blood cells per microliter. You good with that? Okay. Now, obviously, it's going to vary slightly if you're talking about male versus women, if you're talking about adult versus children, but generally, the numbers I want you to kind of remember, and of course, every lab is going to be different, but what I want you to know when it comes to red blood cell lab values, go with the numbers roughly around 4.5 to 6 million red blood cells per microliter. You good with that? All right, let's keep going. Now, when it comes to red blood cells, so right now we're talking about red blood cells. So I just said what the actual lab values are. Tell me that number again. It's right there on the screen. It's going to be 4.5 million to 6 million red blood cells per microliter. All right, let me just erase that so I can have some space on the screen. Now, when it comes to red blood cells, what do they look like? Okay. Well, first, let's back up. What is the purpose of a red blood cell? What's the job of a red blood cell? So, red blood cells, their primary purpose is to be able to carry, for instance, nutrients and carry oxygen. Well, the red blood cell itself, let me back up. The primary purpose of blood is to be able to transport nutrients and, for instance, transport oxygen. But the primary purpose of red blood cells is for oxygen to attach, okay? Because yes, we're trying to transport oxygen, but the oxygen has to hook up to something. So for instance, red blood cells have this special shape that kind of looks like a donut. So if you imagine a donut and it kind of has this kind of concave side on, it's kind of concave on both sides. So if I look at both sides of the donut, it kind of has this little hole in it, right? We call that biconcave, or we say that red blood cells have what's called biconcavity. Okay, so they're biconcave. Now, so here's a red blood cell. And notice how it has this little dip here and here. That's that kind of biconcave shape I'm talking about. The purpose of that shape is to increase the surface area so that we can have these molecules of hemoglobin. Okay. The purpose of the hemoglobin is, for instance, for oxygen here. So let's draw this in. So these little brown pieces here, that's called hemoglobin. And the purpose of the hemoglobin is so that oxygen can hook up to it. Okay? So this is how oxygen gets circulated through the blood. It actually attaches to the hemoglobin. Now, when it comes to the red blood cell, what kind of shape did I say it's supposed to have? It's supposed to be, how do we describe it? It kind of looks like a donut, right? It's supposed to be biconcave or have biconcavity. But what happens if it doesn't form that proper shape? Well, now you're going to have all sorts of problems. So for instance, what's an example of instead of the red blood cell having that nice donut shape, what if it looks like a beach ball? What if it's perfectly round? That's not normal. For instance, what would that condition be called? Where it looks like a sphere. Okay, that's exactly what happens in, for instance, hereditary spherocytosis. So in hereditary spherocytosis, what's happening is you're missing some of the cytoskeleton. You're missing some of the pieces that cause it to form that donut shape or that biconcave shape. So in hereditary spherocytosis, we're missing proteins like spectrin and ankyrin that instead of it forming the donut shape, it actually looks like a beach ball. Okay, that can be bad. Another situation where it doesn't form the right shape is in the setting of sickle cell anemia. So the red blood cells have that kind of sickle shape. So that's what's here. Okay, so the red blood cells have this kind of sickle shape. Well, think about this. This is not a normal shape. So imagine if this sickle cell is going through the blood vessel, it can tear up the blood vessel, right? Because this has kind of a sharp point. So imagine if it's going through a smooth blood vessel with this nice sharp point. It's like a dagger and it just chops up the blood vessel, right? So not only can it cause all kinds of, it can actually damage the blood vessel itself, but for instance, because it's not a nice smooth shape that flows easily, you can actually have stagnant blood that kind of backs up, that forms clots. You can have all kinds of problems where there's an interruption in blood flow. So that's exactly what happens in, for instance, sickle cell anemia. Okay, any questions about any of that so far? And just type your comments in the chat for me. All right, so let's keep going. Now, how do we actually make red blood cells? So I told you what the purpose is. The purpose of red blood cells is, for instance, to have hemoglobin where oxygen can bind to it, and it has that certain shape to increase the surface area, to increase the oxygen carrying capacity. So you have more spots for oxygen to hold on to, right? Because we're trying to get oxygenated blood flow to everywhere it needs to go, all over the body throughout the tissues. So we talked about, for instance, what are the normal lab values? We talked about the shape of it. We talked about the purpose of it. We said what can happen if you don't have enough. But now let's talk about how do you actually make a red blood cell, okay? So let me get some space here. So by the way, what is the name for red blood cells? The term for red blood cells is erythrocytes. Erythrocytes. Okay, let's break that down. We never want to just use words that we don't know what they mean. Erythro means red, cyte means cell. So erythrocytes literally translates to a red cell. And specifically, we're talking about red blood cells. Okay, so red blood cells are also known as erythrocytes. You good with that? Now, how do we make a red blood cell? Okay, so first, what's going to happen is over here in the bone marrow, this is where things start to happen. Now, for instance, inside of it, let's just draw a cell for a minute. Let's say here's a cell. So, of course, we have normally in most cells, they have a nucleus. However, so they have a nucleus and they have a nucleolus. However, red blood cells are what are called anucleated. So they don't actually have a nucleus. So normally, you know how when you're making DNA, normally DNA, for instance, the process of transcription and translation that happens within the nucleus of the cell. So transcription, of course, is where we're going from DNA and we're going to make that messenger RNA copy. And then we're going to eventually translate that. So we're going to go from DNA to RNA, and then we're going to go to proteins. So that process is called transcription and then translation, right? Well, that normally happens, that starts off in the nucleus. Red blood cells don't have a nucleus, so where else is it going to happen? It's actually going to start off in the cytoplasm of the cell, in the cytosol of the cell. So more specifically, though, is it actually starts off, some of this process starts off in the mitochondria of the red blood cell. So what I want to talk about now is how do we make red blood cells, okay? If I were to take a red blood cell, for instance, see how we're just talking about really basic stuff, right? I'm teaching you a very basic concept, but I'm showing you how if you know more about the basics, you're going to know more about everything. I promise you. Just follow along and see how I do this. So we're talking about a red blood cell and how do you make it? So if I take a red blood cell, let's break it down into its smaller components, okay? Let's go back to the screen. So here's a red blood cell, right? I guess I should have made that red, but you get the point. So we're going to say that's a red blood cell. It's got its molecules of hemoglobin. Uh-oh. Now, you just heard me say a word. I said, what word did I say? It has molecules of what? Uh-oh. So it has molecules of something called hemoglobin. Now, let's break that down. Hemoglobin, right? Well, you just hear people say hemoglobin, but hemoglobin is actually broken down into heme and globin. What? That's right. Hemoglobin is made up of heme and globin. Okay. Now, what is hemoglobin though? So, hemoglobin by itself is a protein. Okay. Oh, tell me, what type of protein is hemoglobin? You know how we classify proteins in terms of their protein structure as primary, secondary, tertiary, quaternary, things like that. What's the term for hemoglobin? Anybody remember? Okay, here, let's go over that for a quick moment. So when it comes to proteins, we say that proteins are either primary, secondary, tertiary, or quaternary, right? Well, what's a primary protein? A primary protein basically consists of amino acids. Everybody good with that? Primary protein is just basically a chain of amino acids in a sequence. So that's a primary protein. A secondary protein is this is what happens like when you're making DNA and you have, for instance, the alpha helix of DNA, that kind of helical shape. That's a secondary protein. A tertiary protein is where it kind of takes on this 3D shape. So it's basically a chain of what's called polypeptides together. So these are big structures, but it's like a chain of one type of polypeptide just linked together. And it starts to take on a 3D shape. Hemoglobin is actually a quaternary protein structure. So basically, what that means is you have two or more different types of proteins interacting together. So let's say that again. Hemoglobin is what's called a quaternary protein structure. That a quaternary protein structure is where you have two or more types of different proteins that interact together. Okay, let me prove that to you. In the case of hemoglobin, so if I take one of these little hemoglobin molecules, guys, we're just talking about really basic stuff, but I'm going to show you how if you know more about the basics, you know more about everything. Hemoglobin, for instance, has what are called alpha chains. So it has two alpha chains, and then it has what's called two beta chains. Okay? So hemoglobin, in terms of adult hemoglobin, hemoglobin has 4 subunits, 2 alpha chains, and 2 beta chains. You got that? Okay? Adult hemoglobin has 2 alpha chains and 2 beta chains. However, what do you know about, for instance, fetal hemoglobin? So let's say in a fetus, in a baby. What type of hemoglobin does the fetus have? Type it in the chat. Okay. The fetus has, for instance, instead of 2 alpha chains and 2 beta chains, it actually has, for instance, 2 alphas and 2. Let's put this in a different color. So fetal hemoglobin has 2 alphas and then it has 2 gamma chains. And the way I remember this, I remember being in medical school and I had this hematologist, or I don't know if she was hematology or pathology, but one of my professors who taught the hematology section, I remember her name is Dr. Janice Johnson. And she talked about, for instance, she said that with babies, the way you remember this is babies, you know how they do that baby talk and they say like gamma, gamma, gamma. She said babies have gamma, gamma, goo goo. So the way I remember that fetal hemoglobin contains gamma chains is because of gamma, gamma, goo goo. So fetal hemoglobin has, for instance, alpha chains and gamma chains, but adult hemoglobin has alpha chains and beta chains, okay? See how we have 2 different types of proteins that are interacting together? That's exactly that's the definition of a quaternary protein structure. So that was just some foundational stuff. Let's talk about hemoglobin again. Sorry about my voice. Hang on a second. Okay, so hemoglobin. We said that red blood cells are made up of heme and globin, right? Or they're made up of hemoglobin, and hemoglobin is made up of heme and globin. Okay, so let's talk about how do we make a red blood cell. You ready? All right, let's go. When it comes to, here's a red blood cell. Before we have a red blood cell, of course, we have to make hemoglobin and we have to make heme and globin, right? Well, how do we make heme, for instance? Okay. Well, so starting out, so this is it's actually more than what I'm going to tell you, but this is just kind of a simple overview of it. We're going to start out in the mitochondria and we're going to have something called succinyl CoA. Okay. Succinyl CoA. Succinyl CoA is going to hook up with something called glycine. So glycine, of course, is an amino acid. And these two are going to make something called ALA. That stands for amino levulinic acid. Okay. So what I said is succinyl CoA plus glycine are going to make something called ALA, which is amino levulinic acid. Now, whenever you're trying to make something, we need enzymes to do that, right? Now, what's the purpose of an enzyme? The enzyme, its job is to help the reaction go ahead and speed up. We don't want to take all day. When we need to make some red blood cells, we don't have like weeks. We need it like pretty soon, right? By the way, how long does it take for most red blood cells to be made? Okay. The immature red blood cell, the erythroblast, the immature red blood cell actually starts off in about 1 to 2 days. But the fully mature, so that starts in the bone marrow, and then it actually goes out and gets into the blood, and it fully matures in about a week. So you start making a red blood cell in about 1 to 2 days, but it's fully mature in about a week. And then how long do red blood cells live on average? So they're going to live about 120 days, okay? Let's just recap that. It takes about 1 to 2 days to start making immature red blood cells. They mature in about a week, and then they live for about 120 days, okay? So what I said is we're going to need an enzyme to speed up this reaction. So the way you name enzymes or the name of the enzyme actually tells you a lot about what does it do. So let's say I need to make something called Bob. Well, if I need to make a Bob, I'm going to use a Bob synthase. What? How about the name actually tells you what it does? So whenever you see the word synthase, what we're doing is we're synthesizing or we're making whatever that substrate is. So if you see Bob synthase, now there's nothing called Bob synthase or Keisha synthase or John synthase, but if you needed to make one of those, you could use a synthase. But what if we need to make ALA? What if we need to make amino levulinic acid? Could we use ALA synthase? Yes, that's exactly what we're going to use. So let's pop me back up on the screen. Okay, good, thanks. So if we need to make us, where am I? We need to make right here. We need to make ALA. We're going to use an enzyme called ALA-synthase, okay? So what we're talking about is we're trying to make red blood cells. But first, we got to make us some heme so that the heme can hook up with the globin, make hemoglobin, and then we can make a fully functioning red blood cell. So let's go back over that. We started with succinyl CoA. It hooks up with glycine, and then using something called ALA-synthase, they're going to make something called ALA. Good with that part. Now, what's going to happen is there are actually several more steps in this reaction. I think it's about 8 steps overall. I'm going to speed up the process, but eventually we're going to make this ring structure called a porphyrin. So you may have heard of a porphyrin ring. So we're going to make this ring structure because what we're trying to make is heme. So in fact, let me just move this down a little. Let me move this heme down. Okay. Because what we're trying to make is heme. First, we made this ALA. We're going to go through several more steps, and eventually we're going to make this ring structure called porphyrin, right? But before we can actually make heme, we need to add something else to it. So kind of like you're baking a cake, and you got all these little ingredients, right? but you gotta be able to have all the ingredients to make it taste good. Imagine making a cake without some sugar. That would just be horrible. So, so we can't make heme without a special ingredient, and that special ingredient is iron. All right, let's go back to the screen. So so right here, we made this ring structure called porphyrin. Now it's actually, there's a protoporphyrin and a porphyrinogen. I'm just going to loosely call it porphyrin, okay, just for the sake of this discussion. But before we can make heme, we got to add some iron to it. So let's do that. Let's change my color here. Let's make it here. We'll just make it, actually I want it orange. Let's go with orange. We're going to add some iron to it, okay? Now, how are we going to get this iron inside of this ring? We need another enzyme. Anybody know what that one is? The enzyme we're now going to use to get the iron inside of the ring, that's called ferrochelatase. Okay, what? That's right. It's called ferrochelatase. So the way we're going to add the iron is with an enzyme called ferrochelatase. You say it. Ferrochelatase. You got it? Okay. So ferrochelatase is going to come in and it's going to help us get that iron inside of the ring. And now we have a structure called heme. Okay. Now you just heard me tell you about this enzyme called ferrochelatase, right? What was its job? The job of ferrochelatase is to help the iron get inside of the porphyrin ring so we can make heme. And we need heme to hook up with globin, so we have hemoglobin, and we need hemoglobin so we can make the red blood cells. So what I want you to know is one very vital component of hemoglobin synthesis is iron, okay? So you need iron and you need ferrochelatase to get the iron in there, into the ring. What if you don't have enough iron? What if you have iron deficiency anemia, for instance, okay? Well, how could that happen? Well, it could happen for a number of reasons. For instance, it could be that you could have a gastrointestinal situation that prevents iron from being absorbed properly. You could have some kind of other heavy metal toxicity that competes for the site. So you have other metals that are preventing iron from being able to get absorbed. You could have a viral infection. You could have medications that inhibit it. There's so many reasons. Or it could be something simple like you just don't have enough iron in your diet, for instance. Where do you get iron in your diet? Well, of course, it comes from animal products. So let's say we're talking seafood, poultry, um, uh, animal muscle. Okay. So, for instance, let's say that you're vegan and you're not eating animal sources. That's okay because it's still in vegetables as well. What are great sources of iron that are non-animal sources, so plant-based sources? So that's going to be like, let's say, grains, legumes. It's going to dark green leafy vegetables like spinach, collard greens, things like that. Great sources of iron. Okay. Oh, for instance, or let me ask it this way: what three things are added in prenatal vitamins, for instance? Because, for instance, let's say a woman who's pregnant, one of the things that she can develop is iron deficiency anemia. So one of the things that gets added in prenatal vitamins is iron. What two other things get added into prenatal vitamins? I wasn't planning on going there, but let's go. Okay, so we're just talking about prenatal vitamins. What components make up prenatal vitamins? Number one, we're going to have iron. What else are we going to have? Okay, another thing that we need is calcium. And then another thing that gets added in prenatal vitamins is folate. So folate, of course, is vitamin B9. Okay. Oh, what happens if you don't have enough folate? What do you need folate for? Well, you need folate to make DNA, to make all kinds of cells. If you have a folate deficiency, it can lead to things like megaloblastic anemia. It can lead to neural tube defects, so spina bifida, things like that. All kind of neural tube defects where the baby doesn't actually form properly, or at least the neurological system. So, like things like the brain, the central nervous system, so the brain, the spinal cord, right? So this is how you can have a kid that develops without a brain. Maybe they have anencephaly, or maybe they have like I said, spina bifida or things like that. Obviously, that's not compatible with life. So this is why we want moms to take prenatal vitamins, for instance. But for instance, let's say the mom doesn't even know she's pregnant. So she's not necessarily following her cycles as well, or maybe she has irregular cycles. And so she can actually be several weeks into the pregnancy, still not know it, but the baby has already started to develop, or hopefully. So, for instance, most neurological defects happen, let's say, within the first like 8 weeks, um, or most of the neurological development should happen within the first 8 weeks. So, if the mom is not having enough folate in her diet, for instance, she has a vitamin B9 deficiency in that first 8 weeks, it's going to predispose her to all kinds of neural tube defects. Okay. So, in prenatal vitamins, we have iron, we have calcium, we have folate. Okay. Now, let's go back to, remember how I was telling you about this? So you see what I'm doing here. I'm taking little basic things, right? And just because I know very basic topics, we're able to go all over the place just with a basic idea. And this is how I want you to be. I want you to be able to take very basic concepts. And when you know the biochemistry, when you know the physiology, you're going to be able to figure out everything else very quickly. Okay. Let's go back. Remember how I said that the enzyme, what was that enzyme called that helps us get the iron into that porphyrin ring? That was called ferrochelatase, right? Well, there is a condition where, remember, so we just talked about if you have an iron deficiency. So we needed iron, but we also need the enzyme to help the iron, and that's ferrochelatase. But what if you have a ferrochelatase deficiency? Do you know the name of the condition where you don't have enough of that enzyme? Do you remember what that's called? So here, let me get some more space here. So a ferrochelatase, or the condition is called EPP. It stands for I'm going to break it down. Erythropoietic porphyria. Okay, that's erythropoietic protoporphyria. What is that? That was a mouthful. Let's break it down. Erythro, that deals with or erythropoiesis is the word for production of red blood cells. So making red blood cells, the noun form of that is erythropoiesis. The adjective form of that is erythropoietic. Okay, so that whole word just means something involved with making red blood cells. So erythropoietic, and then protoporphyria. Remember how we were talking about you need this ring structure called this porphyrin ring? Well, as a part of this is something called porphyrin. So what happens in this situation, in this condition called EPP, or erythropoietic protoporphyria, what happens is you're missing the enzyme. What does it do? It prevents you from being able to get the iron into that ring structure. So what happens is porphyrin starts to accumulate. So it doesn't go all the way to making hemoglobin, for instance. Before it makes it all the way to the finished product, you get these other things that kind of accumulate. So for instance, porphyrin can actually accumulate into the skin. And for instance, let's say they go outside in the sunlight. Now the sun hits their skin, it causes an enzymatic reaction underneath their skin. Now they develop a rash, for instance. That rash can be painful, so they can have photosensitivity, especially with sunlight and the UV rays. The porphyrin can also get deposited, for instance, in the liver. It can cause all kinds of problems. So that's a condition where you don't actually have that enzyme. Okay.